Alright, so for this video, we'll be looking at two particular examples of epistasis. The first is recessive epistasis. Okay, we'll go straight to the example in the notes, right? So this example involves the coat color of a dog, right? Labrador Retriever. So the question very clearly tells you, right, that this one phenotype, coat color, is determined by two genes, B and E. Right, so straight away, gene interaction. For gene B, right, the dominant allele codes for black, the recessive allele codes for brown, and for gene E, the dominant allele allows the color pigment to be deposited in skin cells, and the recessive allele, right, prevents this deposition of pigments. Okay, so for the recessive E, right, it gives a golden color because of the lack of deposition of pigments. So you have the cross between the purebred black dog with the double homozygous recessive golden dog, right? F1 were all black, and then you're supposed to find the F2 ratio. Okay, so just to summarize the information given in the question, right? Big B allele codes for black pigment, small b allele codes for brown pigment, big E alleles allows for the deposition of pigment, and the small e allele prevents the deposition of pigment and therefore gives a golden color. All right, so just to test our understanding, right? If I give you these four different genotypes, right, what would be the phenotypes according to the information given, right, in this question? Right, so the first genotype, a double heterozygote, right, it would be black in color, right, because the big E, okay, follow my cursor here, the big E allele, right, allows for deposition of the black pigment due to the big B allele, so the dog is black. For the second genotype, it would be brown because you have brown pigment which is allowed to be deposited by the big E allele, right? For the genotype number 3 and number 4, both will be golden or pale golden. It should be golden according to the question. Okay, so both would be golden because even though there is the expression of black pigment or expression of brown pigment, they are not allowed to be deposited due to the small e genotype, right? So both are golden color. So this is an example of the recessive epistasis because the homozygous recessive genotype of gene E, in this case, mask the expression of gene B, right? So regardless of whether the gene B genotype is heterozygous, homozygous dominant, homozygous recessive, the same phenotype of golden color is expressed due to the homozygous recessive small e, right? So the homozygous recessive genotype of gene E mask the expression of gene B, right? So therefore recessive epistasis. Okay, so looking at the genetic diagram, right? How do we draw a genetic diagram? It is pretty similar to a normal dihybrid diagram, right? So we will start off with the parental according to the question. Black crossed with golden, right? The question tells us the black is purebred, right? So the only possible genotype is double homozygous dominant. The question tells us the golden color, the golden dog is double homozygous recessive. Right, so same thing that you do for a normal diagram. You have meiosis and gametes, right? Then you cross them to get F1, which are all black, as shown here and also mentioned in the question. Then you have one F1 male crossed with one F1 female, so the siblings cross, I suppose it's okay with dogs. Okay, so anyway, meiosis and the 4 different types of gametes, right, for each of the parent. Then the Punnett square, the 4 different classes of genotypes, right, possible for the F2. So far similar to a normal dihybrid diagram, right? But the difference is in the F2 phenotype, right? Because the small e genotype would mask the expression of the gene B, right? So regardless of the genotype for gene B, whether it is homozygous dominant or heterozygous or homozygous recessive, right, the same phenotype of golden color is expressed. So therefore, these two classes of genotype gives you the same phenotype, and the ratio, instead of 9:3:3:1, becomes 9:3:4 because the 3 and 1 added together, right? So 9:3:4 ratio for the F2. Right, so this will be the answer to the question earlier in the example given. All right, so this is how to draw a recessive epistasis kind of genetic diagram. Okay, so again, right, the two different classes of genotype giving you the same phenotype due to the recessive epistasis that we have looked at earlier. Okay, so just a bit of a summary of what we have looked at, right? It is epistasis, gene interaction, right? So two genes determining one phenotype as part of gene interaction. And the type of interaction, the type of epistasis, is when the recessive genotype of one gene masks the expression of the other gene. That's why it's called recessive epistasis. Epistasis because it's the recessive genotype of one gene that masks the other gene. Right, so recessive epistasis. Okay, so when a double heterozygote is crossed with another double heterozygote, instead of 9:3:3:1, you get 9:3:4. So this is what it means to have a reduction, right, or decrease in the classes of phenotype that we have mentioned in as a general feature of epistasis in the first video. Okay, so in a normal dihybrid, right, for the F2, there should be 4 different phenotypes, right? That's why you get 9:3:3:1 ratio, right? But in this case, there are only 3 classes of phenotype because the last 2 in the diagram, right, the last 2 classes of genotypes give you the same phenotype due to the masking effect by the small e genotype. So you only have 3 classes of phenotype and therefore a 9:3:4 ratio. Okay, so that would be an example of epistasis, right? Recessive epistasis. Next, we will look at another example of epistasis known as dominant epistasis. Okay, so again, we'll start off with the example given in the notes where we have this squash plant, squash fruit color. So again, right, one trait, one phenotype determined by 2 genes. So another, very clearly, another example of gene interaction. So you have the one gene, gene D, right, with the dominant allele that suppresses the expression of the other gene, gene G, right, leading to white fruits. Right, so this question is very clear, telling you the kind of epistasis, right? The dominant allele of one gene, gene D, mask the expression of the other gene, gene G. Right? That's why it's called dominant epistasis, because it's a dominant allele of one gene that masks the other gene. Okay, so anyway, then small d, allele small d, no effect. Right? So gene G, which codes for green pigment, and rather the dominant allele G codes for green pigment, and the recessive allele G codes for yellow pigments. Okay, so you have white fruits, plant with white fruits crossed with plant with yellow fruits. F1 OR produces white fruit, and the selfing of F1 produces white fruits, green fruits, and yellow fruits. Right, so all the possible phenotypes are expressed in the F2 that tells you that F1 should be a double heterozygote. Right, so with that clue, right, F1 being double heterozygote, right, you can then deduce the genotype of the parents, which I'll show you in the diagram. Right, so anyway, before we go to the diagram, right, again, just to understand the mechanism of this epistasis, right, so can you deduce the phenotypes for these 4 genotypes? Okay, so the first one, heterozygous for D and homozygous dominant for G, that would give you white color because of the dominant allele D, right, which masks the expression of gene G. Then you have white again, right, because of the dominant D again. Right, so for the recessive D, now gene G can be expressed. Right, so the dominant G gives you the green pigment and therefore green color. And the last one, right, recessive G gives you the yellow color, which is expressed due to the small d genotype, right? So there's no masking. All right, so in this case, the big D, right, whether it is a homozygous dominant or heterozygote, we would mask the expression of gene G, and therefore this is known as dominant epistasis, because the masking is by the dominant allele of one gene, right, which masks the other gene. Okay, right, so this is the diagram that you have right in the notes. Okay, so the again, right, you have the observed number given, so I go backward because the clue given, right, is kind of backward. Right, so you are given the observed numbers, which kind of is similar to the ratio 12:3:1. Okay, and we know that, right, the genotypes that have got big D, regardless of the genotypes for G, would give you white fruit, right? And then the green fruit for the small d, big G, and the yellow fruit for homozygous recessive for both genes. Okay, so instead of the 9:3:3:1, right, the 9 and 3 is added together, and therefore it's a 12:3:1. All right. And I said earlier, right, because you have all the phenotypes possible shown in F2, so that means that your F1 should be a double heterozygote. And the question says that the F1 is self, right? So you have double heterozygote crossed with double heterozygote. That's why it should be 9:3:3:1, but then the 9 and 3 is added together to be 12. Okay, and in order for you to have double heterozygote, right, then these are the two parental genotypes that is possible, right? So for the yellow fruit, right, because it must be small d, right, and it also must be small g in order for it to be yellow, right? So you know that this must be a double homozygous recessive. So in order to get F1 to be heterozygote or double heterozygote, the other parent must be double homozygous dominant, which will give you white, right? So it follows the clues given in the question, right? So the answer to the question would be these two genotypes, right, for the parents. Okay. So anyway, for the dominant epistasis diagram, again, it is very similar to a normal dihybrid diagram. The only difference is when the F2 phenotype is shown, right? You have two different classes of genotypes, right, coding for the same phenotype due to the dominant epistasis. Okay, right, so just to summarize again, for the dominant epistasis, is the still gene interaction, right? So two genes coding for one phenotype. Unlike the recessive epistasis, right, in this case, because it's dominant epistasis, right, so it is a dominant genotype of one gene masking the other gene, right? So instead of 9:3:3:1, you have 12:3:1. Okay, right, so that would be recessive and dominant epistasis, two examples of epistasis.